Hello students, welcome back after the break. So we were discussing about reversion and suppression. As a first part of my lecture, I have discussed about two different forms of reversion, that is true reversion and equivalent reversion. So what is true reversion? True reversion, as a result of true reversion, that is a form of back mutation. The mutant is reverted back to the wild type, not only in terms of its phenotype, but also in terms of its genotype. Now, let us consider a very simple example, review a very simple example of true reversion, although I've discussed about that example in my previous lecture. Now, supposedly AAN, consider a mutation in that is mRNA codon AAA, which codes for amino acid lysine. It's mutated to GAA as a result of forward mutation. The GAA mutated codon codes for amino acid, that is glutamic acid. Now, the mutant GAA experiences a reversion or back mutation, as a result of which it is again mutated to AAA, that is which again codes for lysine. So, as a result of this, what are the effects of this kind of back or reversion? That is, AAA is restored back to AAA, both in terms of its mRNA codon sequence and in terms of its encoded amino acid, that is lysine. So such a reversion is known as true reversion. And what is equivalent reversion? Often it happens that the mutant, as a result of back reversion, is mutated back to the wild type, but only the phenotype of the wild type is restored, but the genotype is not restored. Now, in that case, the reversion is known as equivalent. Sorry, the previous reversion was an example of true reversion. In that case, in this case, the reversion is a type of equivalent reversion. Now, let us take the example of a codon coding for an amino acid, that is mRNA codon, say UCC, which codes for amino acid serine. As a result of forward mutation, it's mutated to another codon, that is UGC, which codes for amino acid cysteine. But again, now this experiences a back or a reverse mutation, and this UGC is mutated to AGC, to a synonymous codon, which again codes for amino acid, that is serine. So what happens in course of this back mutation? That is UCC, that is which codes for serine, as a result of back mutation is mutated to a codon called AGC, or a synonymous codon, which again codes for serine. So the phenotype is restored as a result of back mutation, but there's a difference in mRNA codon and the sequence of the mRNA codon, that is gene, and hence the genotype is not restored. So this is an example of equivalent reversion. Now, before I went for the break, I left a very interesting question for you all. Just think, you might have thought about it. What is the use of this reversion in genetics? Therein lies a huge or a tremendous application of reversion in genetics. Now, reversion is generally used to direct or to make a bacterium revert back to its original type. Now, so let us supposedly say that a bacterium is not able to tolerate lactose as a result of forward mutation. Now, we can make the bacterium revert back to its ability to tolerate lactose, or the bacterium regains its ability to tolerate lactose when it's grown in a medium containing lactose as the only source of carbon and energy. So this is what is called as directed or adaptive mutation. So what is the use of this, and what is actually happening in this case of directed or adaptive mutation? It is nothing but a form of reversion in which the mutant bacterium is reverted back to its wild type, as a result of which it regains its ability to use lactose. Hope it's a bit clear to you about that. You can a bit clear to you that reversion can be used or has a tremendous application in genetics. Now let us study about suppression. what is suppression? Suppression is a second type of mutation which suppresses the effect of the first mutation. The phenotype is only restored, but the genotype is usually not restored as a result of suppression. Hence, suppression is nothing but a second type of mutation which suppresses the effect of first mutation. That is known as suppression. Now, there are again two different forms of suppression depending upon the location or depending upon the position in which the suppression occurs within the sequence of DNA or within the same gene, or it occurs in a different gene. Often, suppression in the form of second mutation occurs within the sequence of the gene or occurs nearby the gene. So, such a suppression is known as intragenic suppression. So, it is nothing but a second site reversion. Now, as a result of intragenic suppression, the phenotype is restored, or the nearby phenotype of the protein is restored. Now, let us consider an example. Let us take a sequence, DNA sequence, CAT, CAT, CAT. As a result of forward mutation, as a result of insertion in the forward mutation, an X, that is a nucleotide, say X, gets incorporated and the mutated sequence becomes CAT. So what is there is a change in the codon reading frame, that is CAT, and likewise. And now again, if this X is removed as a result of suppression or second mutation within the same locus of the gene, what happens? The gene that is the original genotype is restored, and when the genotype is restored, it's not only the genotype which is restored, but the phenotype is also restored as a result of suppression. And as a result of removal of the nucleotide X, which has been previously added as a result of forward mutation, the sequence reverts back to the original sequence present at the locus of the gene, present at the region of the gene, that is CAT. So such a suppression or a second mutation which suppresses the effect of first mutation and reverts back not only the phenotype but also the genotype occurring within the same region of the gene is known as intragenic suppression. So now, what is the use of intragenic suppression? There are multiple uses of intragenic suppression. Intragenic suppression is used to study the structure-functional relationship between a protein. Why? Because it is very tightly linked to original form of mutation. So you can study more about the original form of mutation following an intragenic suppression. That is mutating an amino acid sequence, that is to a different amino acid sequence via addition, and by deletion again reverting back to the same amino acid sequence or reverting back to an amino acid that is sequence of a protein which is nearby similar in terms of its amino acid content. So that is the use of intragenic suppression. But what is there? Intragenic suppression actually doesn't serve the purpose of suppression hunt. Why is it so? Because intragenic suppression does not generate new proteins as such. As a result of intragenic suppression, the genotype is restored, and because of intragenic suppression, a protein that is the result of the suppression is a protein which differs hardly in few amino acids and is almost same as that of the original protein, or is similar in structure and function to the original protein. Hence, it does not serve the purpose of suppression hunt. So, which form of suppression serves the purpose of suppression hunt? Another form of suppression which serves the purpose of suppression hunt is known as intergenic suppression or extragenic suppression, when there is a second form of mutation which suppresses the effect of first form of mutation, but it occurs in a different different region of the gene, or in a different region of DNA of the gene, or particularly or exactly, that is exclusively in a different gene, it's known as extragenic suppression, or it is also known as intergenic suppression. So supposedly, let us that is, I'll explain it more with the help of an example. Say, as a result of as a result of mutation and a relationship between a stable interactive relationship which involves interaction between a group of proteins is lost. Now, by intergenic suppression in a different region of the gene, in a completely a different region of DNA, that is in a different gene, this function can be somewhat restored, and the new protein can be generated, and the interactions between the protein can be somehow restored back. So this is a very important application of intergenic suppression. That is, it's used to generate new proteins, and it's used to study the expression of proteins which are involved in signal transduction, which are the regulatory proteins, all involved in transcription, translation, and of course in many different biochemical pathways. So we have all come across, to summarize, we have come across different forms of suppression and reversion. Reversion is nothing but a back mutation, as a result of which a mutant is reverted back to its original wild type. If the mutant is reverted back both in terms of its genotype and phenotype, it's known as true reversion. If the mutant is reverted back only in terms of its phenotype, but the genotype differs, it's known as equivalent reversion. Now, there are two forms of suppression. Suppression is nothing but a second mutation which is used to suppress the effect of first mutation. And if that second mutation occurs within the same region of the gene or DNA region, or nearby the gene, that is the sequence of the DNA, it is known as intragenic suppression. As a result of intragenic suppression, the wild type function is restored and a protein is generated which is similar to the wild type, or is almost the wild type protein, or differs from the wild type in one or two amino acids. So it's used to study the structure function relationship between different proteins, but it does not produce variation or new proteins as such, and that does not serve the purpose of, that is, a suppression hunt, and generates the need for another form of suppression. So next to that, a new form of suppression which came into advent and which comes into effect is known as intergenic suppression, in which suppression occurs totally in a different gene or in a different region of DNA. And as a result of this, what happens? A new protein is, while the suppression occurs in different region of the DNA, different region of the DNA, or different region of the gene, or in a different gene, say different locus of the gene, a new protein is generated. So supposedly, I explained it by taking an example of protein-protein interactions. If a mutation disturbs a protein-protein interaction as a result of intergenic suppression or extragenic suppression in a different, so-called extragen, also called extragenic suppression, in a different gene, a new protein can be generated which will restore somehow the protein-protein interaction, that is, which will somehow restore that function, and it's used to generate a series or an area of proteins, and is used to study, it's well applicable for the proteins which are involved in signal transduction pathways, regulatory proteins involved in transcription, translation, and many more things. So that is the end of my lecture meant for today. Now, hope the basic concepts of reversion and suppression are very clear to you. Now, at this point, I leave a very activity corresponding to my today's lecture. It is that think about the application of reversion and suppression in genetics, and what is the need of intergenic suppression. Then, why is intergenic suppression replacing intragenic suppression, or the suppression in a different gene, is preferred in response to, or preferred more than, the suppression within the same gene, which is called intragenic suppression. So there are many more questions, many more facts about suppression and reversion which are yet to be unfolded, which I'll be unfolding in my upcoming lectures. So stay healthy, study, know about suppression and reversion from basic book, that is book of microbiology, broad biology of microorganisms. You may consult the general book of microbiology by Prescott, Harley and Klin, and so on. So stay tuned for till I come up with another new lecture of microbial genetics. So goodbye, have a nice day, goodbye to all my students.